are the enzymes that cleave nucleic acid. These enzymes are capable of cleaving phosphodiester bond between nucleotides of nucleic acids. Nucleases belong to a class of enzyme known as hydrolase. Nucleases can be divided into two different groups. One is deoxyribonuclease or DNase that cleave DNA. The other group is ribonuclease or RNase that cleave RNA. Nucleases can also be categorized into exonuclease and endonuclease. An exonuclease is an enzyme that cleave off nucleotides one at a time from the 3' end and 5' end of DNA and RNA chain. An endonuclease is an enzyme that produces internal cuts. That means endonucleases cleave internal phosphodiester bond. There are many endonucleases which cleave only one strand of a DNA duplex. For example, S1 nuclease, which has been isolated from Aspergillus oryzae. There are many endonucleases which cleave both the strands of a double-stranded DNA molecule but at a random sites. There is another group of endonuclease known as restriction endonuclease. Restriction endonuclease is an enzyme that recognizes and cleaves both the strands of a double-stranded DNA molecule within or near specific recognition site. Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about restriction endonuclease. The name restriction endonuclease has been given on the basis of a phenomenon known as host restriction. What is host restriction? A stock of lambda phage grown in one strain of E. coli, for example strain B, infect that strain with high efficiency, but the same stock of lambda phage infect another strain of E. coli, for example strain R, with low efficiency. This phenomenon is known as host restriction, and this phenomenon was characterized by Arbor, Smith and Nathans in 1978. In 1978, Arbor, Smith and Nathans were awarded Nobel Prize for the discovery of restriction endonuclease. So, the host restriction is a bacterial analog of immune system that allow bacteria to recognize and to defend themselves against foreign invader. Naturally occurring restriction endonucleases are divided into 5 different groups on the basis of composition, enzyme cofactor requirements, nature of the target sequence and the position of the cleavage site relative to the recognition site. The 5 different groups of restriction endonucleases are type 1, type 2, type 3, type 4 and type 5. First of all, we shall discuss about restriction endonuclease type 1. Type 1 restriction endonucleases where the enzyme first to be identified, and for these enzymes the cleavage site is at a distance of at least 1000 base pair away from the recognition site. The recognition site is asymmetrical and it is composed of 2 specific portions. One portion contains 3 to 4 nucleotides and the other portion contains 4 to 5 nucleotides. Between these two portions there is a non-specific spacer that contain 6 to 8 nucleotides. Type 1 restriction endonucleases are multifunctional enzyme that has two activities. One is restriction digestion activity. And the other is restriction modification activity. The type 1 restriction endonucleases require S-adenosine methionine. Type 1 restriction endonucleases require S-adenosine methionine, hydrolyzed ATP, and divalent magnesium ion for its full activity. And type 1 restriction endonuclease has multiple subunits. The subunits are HSDR, HSDM, and HSDS. HSDR is required for restriction digestion activity. HSDM is required for restriction modification activity, and HSDS subunit is required for specificity of the recognition site and also for restriction digestion and restriction modification activity. This is about type 1 restriction endonuclease. Type 2 restriction endonucleases are most commonly available and commonly used restriction endonucleases. They form homodimer. The recognition site is undivided and palindromic, and the recognition site is 4 to 8 nucleotides in length. Type 2 restriction enzymes require divalent magnesium ion for their activity, and these enzymes recognize and cleave at the same site. The cleavage by type 2 restriction endonucleases eat two types of ends. One is blunt end, and the other is cohesive end. Or sticky end. This is about type 2 restriction endonuclease. Type 3 restriction endonucleases recognize two separate non-palindromic sequences that are inversely oriented, and the cleavage site is present at least 20 to 30 base pair away from the recognition site. Type 3 restriction enzymes are heteroolygomeric and multifunctional. These enzymes require ATP but do not hydrolyze it. S-adenosine methionine stimulate the rate of reaction but is not required. And these restriction enzymes exist as a part of a complex with a modification methylase. And type 3 restriction enzymes are component of prokaryotic DNA restriction modification mechanism that protect the host against foreign DNA. And type 3 restriction enzymes consist of two subunits. One subunit is mode subunit, and the mode subunit recognize DNA sequences. Mode subunit recognize DNA sequences, and it is a methyl transferase. And another subunit is rest subunit. Rest subunit is required for restriction digestion. Rest subunit is required for restriction digestion, but it has no enzyme activity on its own. This is about type 3 restriction endonuclease. Type 4 restriction endonuclease. Type 4 restriction enzymes recognize methylated DNA. And last one is type 5 restriction endonuclease. Type 5 restriction endonuclease utilize guide RNAs to target specific non-palindromic sequences that are present on the invading organisms. This is about different types of restriction endonucleases. Applications of restriction endonucleases. Restriction endonucleases are used to manipulate DNA for various scientific applications. Restriction enzymes are also used to assist in the insertion of DNA into different types of cloning vector during cloning and protein production experiment. By using restriction endonuclease, we can cleave different DNA fragments at a specific restriction site. So they are very useful in cloning and protein production experiment. Restriction endonucleases are also useful to distinguish gene alleles by recognizing single base change, which is known as single nucleotide polymorphism. Restriction endonucleases are also useful to digest genomic DNA for the process of southern blot. Hello friends, this is about restriction endonuclease and different types of endonuclease and their applications. Please share, like and subscribe this video, and also please give your comment